So today we are going to talk about the cell cycle. The cell cycle is an orderly set of stages from the first division of a eukaryotic cell to the time the resulting daughter cells divide. Just prior to the next division, the cells grow larger, the number of the organelles double, and DNA is replicated. There are two main or major stages of the cell cycle. There is interphase, which has a couple of different subcycles, and there is the mitotic stage, and this includes what we call mitosis and cytokinesis. So today we are going to go through these, and we are going to talk about what is happening in each of these phases and what happens if something goes wrong in one of these phases. So the first phase is interphase, and most of the cell cycle most of the time happens here. So about 90% of the cell cycle is actually spent in the phase of interphase because there is a lot going on here. So, nerve and muscle cells do not complete the cell cycle, and they always remain in what we are going to find out is called G0. So, interphase consists of a G1, an S, and a G2 phase. And like I said, it takes up about 90% of the time that we find a cell in the cell cycle. And there are distinct things that are happening in each of these phases. So in the G1 phase, we see a cell recovering from the previous division. We see a doubling of organelles. We see a growth in size. And we see an accumulation of raw materials so that more DNA can be synthesized. In the S phase, DNA replication takes place. Proteins associated with DNA are synthesized. Chromosomes enter the S phase with one chromatid each, but they leave the S phase with two identical chromatids, or what we call sister chromatids. Sister chromatids remain attached until they are separated during mitosis, and we will see that in a bit. The G2 phase is located between the DNA replication and the onset of mitosis. The cell synthesizes proteins, including microtubules, that are all necessary for the cell to divide. The M or mitotic stage is what comes next, and this includes mitosis and cytokinesis. Mitosis is the division of the nuclear material. Daughter chromosomes are distributed by the mitotic spindle to two daughter nuclei. So it is the splitting of the chromosomes. Cytokinesis is the division of the cytoplasm. And this process results in two genetically identical daughter cells. So how is the cell cycle controlled? Well, a signal is a molecule that influences the activities of a cell, and the cell cycle is controlled by both internal and external signals. An example of an external signal are growth factors that signal proteins that are received at the plasma membrane. Internal signals might be the family of proteins called cyclins, and they increase and decrease as the cell cycle continues. There are specific cyclines that must be present for the cell to proceed from one stage to the next. So if these signals, these internal signals, are not present, the cell cycle will stop at G1, M, or G2, which we call checkpoints. So basically, your body, your cells are making sure that this cell cycle is going as it should. And if something is detected to be incorrect or wrong, it will hopefully shut that cell down. Because we don't want any cells that are doing their own thing, right? We want them to be replicating correctly with the correct amount of chromosomes and the correct amount of proteins and whatnot. Apoptosis is programmed cell death. And this involves a sequence of cellular events that bring about the destruction of the cell. And you want this to happen. You want your body to be kind of eating up or destroying the bad things that they might have in their cells. So this is a good thing. And apoptosis can be triggered by different enzymes, and some of these are called caspases. And these literally come in and start to destroy things that your cell doesn't need. So mitosis and apoptosis are basically opposing forces, because mitosis will increase the cell number by dividing, by division, and making more cells, whereas apoptosis is decreasing the cell number because it is eating up things you don't need and destroying bad cells. Apoptosis and cell division occur throughout our lifetimes and basically allow our bodies to remain homeostatic, because hopefully they are balancing one another out correctly, making what we need through mitosis, getting rid of what we don't need through apoptosis. So if there are abnormalities in cells, these cells could become cancerous, right? Or maybe they are infected with a virus. And we are hoping that our body comes through and uses apoptosis to destroy those bad cells. Okay? So that's what we're talking about here. There is a signaling protein, which is referred to as P53, and this will stop the cell cycle at G1 when the cell's DNA is damaged. So again, another checkpoint that your body has. If there's something wrong with the DNA, we don't really want that cell being made. So it will be shut down because of this protein P53. That might also initiate an attempt to repair the DNA, and if it is repaired correctly, then the cell cycle can continue and keep going in mitosis. But if it cannot be repaired, your body will tell apoptosis to come in and get rid of that cell, clean it up. We don't want it. So, human cells are eukaryotic, okay? And we have chromosomes. So we have a single DNA molecule, which is a double helix, or sometimes referred to as a twisted ladder. And DNA is associated with histones. And histones are proteins. And DNA and histone proteins are collectively called chromatin. Histones play a structural role. They have essential survival functions. There are primarily 5 types of them, and they are responsible for DNA packing into the nucleus. The DNA is wound around an 8 histone core that is called a nucleosome, and this appears to look like a bead. Nucleosomes are joined together by what we call a linker in the DNA, and this appears as beads on a string. That's why it looks like that. The string folds into a compact zigzag structure. And then it loops into radial loops, and it is loosely coiled. Euchromatin represents the active chromatin that can be transcribed by RNA polymerase and transcription factors. So that is part of when the DNA is replicating. A more highly compacted form of the chromosome is called heterochromatin, and this is inactive chromatin. And the genes here are hardly ever transcribed. Compact chromosomes move more easily moved than extended chromatin. And most chromosomes have both compaction levels. So, again, mitosis is the nuclear division. Cytokinesis is the division of the cytoplasm. Two distinct phases and divisions. During mitosis, sister chromatids are separated and distributed into the daughter cells. But before mitosis can begin, the chromatin needs to condense and coil into distinctly visible chromosomes. And each species has a characteristic chromosome number, meaning each species has, a set number of chromosomes. Humans have 46. So, the diploid or 2N number includes the two sets of chromosomes of each type. So like I said, humans have 46 chromosomes. So our diploid number is 46. Haploid refers to the number of half of, 2N, which was our diploid, just N is half. So haploid is half of the number of chromosomes, or in humans, that would be 23. So basically, humans have 23 different types of chromosomes, and we have two of each, right? Because we get one from our mom and one from the dad, right? We get two sets of chromosomes. When the sperm and the egg meet to make the zygote, they are each hopefully bringing 23 chromosomes, and they all match up. And that diploid number gives us that 46. Important to know these numbers. Haploid is just the N, for humans it's 23. Diploid is 2N, and for humans it's 46. So, during interphase, the cell is preparing for cell division. Organelles are duplicated, including a centrosome. At the end of the S phase, each chromosome internally has been duplicated. So it consists of two identical double helical DNA molecules. We have what are called sister chromatids, two strands of genetically identical chromosomes. And they are attached in the middle at what we call a centromere. So sometimes they look like little X's. During mitosis, that centromere that is holding those sister chromatids together is going to separate, so that those sister chromatids separate. And they each become a daughter chromosome. And then each of those daughter chromosomes are distributed to one or the other daughter nuclei. So the information is split. So basically you have a normal cell that duplicates, and then it cuts its information in half and makes two new cells. Then those cells duplicate their information and make two new ones out of themselves. Do you understand? Like, it's a continuous process. So, just outside the nucleus is the centrosome. This is the microtubule organizing center in animal cells. The centrosome is also replicated in the S phase of interphase, so there are 2 centrosomes before mitosis begins. And this allows for that split so that each chromatid can go to a new cell. The centrosome organizes on what we call the mitotic spindle. This spindle contains many fibers, and the centrosomes then kind of hook up to it so that they can all get in an orderly manner in order for division to take place. So, prophase is the first phase of mitosis, and this is where we are going to see the nuclear division take place. The chromatin here is condensed, and if you're looking through a microscope, you can actually see the chromosomes because they've condensed down. Each chromosome has 2 sister chromatids that we said are held together at the centromere. The nucleolus starts to disappear, and we see some nuclear envelope fragments. And you can start to see this spindle starting to assemble. The 2 centrosomes start to move away from each other, and in animal cells we see the microtubules form a star-like array which we call asters. And these are kind of at the end. The spindle kind of looks like a football, if you will, and the asters are at the ends. We then have prometaphase, and this is a preparation for the sister chromatids separation that is coming. The centromere of each chromosome develops 2 kinetochores, and the kinetochores are specialized. They're a specialized protein complex, one attached to each sister chromatid. We then have metaphase. In metaphase, the chromosomes are pulled around by these ketochore fibers. They are forced to align across that equatorial plane of the cell, or what we call the metaphasal plate. And this represents a plane through which the mother cell will be divided. There is a non-attached polar spindle fiber that we see, and it overlaps. And the M checkpoint delays the start of anaphase, which is the next phase, until all the kinetochores are properly attached or ready to go. In anaphase, we see the centromere dissolve, and therefore the sister chromatids are released. And they each separate at that centromere. They are now called daughter chromosomes, and they are pulled to the opposite poles along those kinetochore fibers. And the poles move farther and farther apart. So we are separating that original X that we had, so we now have 2 daughter chromosomes so that we can basically have 2 new cells in the end of this process. In telophase, the spindle completely disappears. There are now new nuclear envelopes that form around these daughter chromosomes. There are now 2 clusters of daughter chromosomes. There are still 2 of each type with all types represented. And then you have this nuclear envelope that's forming to start the formation of each new cell. And then we have to have that cytokinesis take place because we also have to split the cytoplasm. So, cytokinesis is the division of the cytoplasm. It allocates the mother cell's cytoplasm equally into each of these new daughter cell nucleuses, into the new daughter cells. It encloses each daughter cell in its own plasma membrane. And this begins in anaphase. We start to see this process coming to be. But it is different in a plant and an animal cell. It is not the same. So in an animal cell, in animal cytokinesis, we see what's called a cleavage furrow. And this appears between the two daughter nuclei. And it is formed by a contractile ring of actin filaments. It's almost like pulling on a drawstring. And it will eventually close in and pinch the mother cell into two. And then pop, you've got two identical cells there from what once was one. Cytokinesis in a plant cell, however, is different because a plant cell has a cell wall. Animal cells do not. So we see a little bit different of a process here. We see what is called a cell plate. So because of those cell walls, we can't see that cleavage furrow. It's a different process. Okay? You're still getting your two cells in the end, but because of that cell wall, you have a different process. Why do our cells go through mitosis? Well, there are several functions. It permits growth and repair. And it allows plants to make more tissue and to increase their girth, which we see when it's growing season, right? You've seen the rings of a tree. Those rings of that tree, and that tree is getting bigger and bigger through mitosis. In mammals, mitosis is necessary when a fertilized egg becomes an embryo and an embryo becomes a fetus. It also is occurring after birth, as a child becomes an adult, or when we break a bone and we need to mend it. It's kind of always going on in animals because for the most part, we're kind of always growing or mending something. So, stem cells. Many mammalian organs contain what we call stem cells, and they retain the ability to divide. Red bone marrow stem cells divide to produce various types of blood cells. So I'm sure you've heard of stem cell research, or maybe you know something about it, but therapeutic cloning is used to produce human tissues, and they can begin with either an adult stem cell or an embryonic stem cell, and you can grow these and actually make tissues. And embryonic stem cells can be used for reproductive cloning, and that is the production of a new individual. So, earlier I mentioned that we want all our cells to be in line and doing what they're supposed to be doing. But we all know that sometimes that doesn't work out like it should. And sometimes we see tumors. So a tumor is an abnormal growth of cells. A benign tumor is not cancerous. It is encapsulated and it does not invade its neighboring tissue, and therefore it does not spread. Whereas a malignant tumor is cancerous. It is not encapsulated and it wants to invade its neighboring tissues. It can also detach from wherever it started and move around. And that is called metastasis. It metastasizes. It moves. And this can result from a mutation of our genes within regulating that cell cycle. If we are not regulating the cell cycle correctly, it can go out of control and it could make a malignant cancerous tumor. The development of cancer tends to be gradual and multi-step, and it could take years before a cell is obviously cancerous. Some characteristics of cancer cells: they lack differentiation, they are non-specialized, and they are immortal. So they just can keep entering that cell cycle repeatedly, which is why and how tumors can grow. They have abnormal nuclei. They can become enlarged, and they can have an abnormal number of chromosomes. And they can have extra copies of genes because, again, they weren't duplicated correctly. Somehow that error wasn't caught in the cell cycle. And now they've got all these things going on in the cells that shouldn't be. They're literally out of control. And they do not undergo apoptosis, which is why cancer doesn't cure itself. Normally, we have our cells, our body coming in and saying, we need to get rid of this damaged DNA. We're undergoing apoptosis. That doesn't happen in cancer. So cancer doesn't just fix itself. Cancer cells are abnormal, like we said, and again, they fail to undergo apoptosis. Double problem, right? Because now they're not going to fit. Now that we've got a trouble cell, we've got a cell with bad information, and it's not going to fix itself. So we've got a double situation. And this forms tumors. So mitosis is normally controlled by contact with neighboring cells, what we call contact inhibition. Cancer cells have lost that. And like we said, they've lost differentiation. So they are non-functional in their tissues. They have enlarged nuclei, and they can move around. They can break into fragments, move to other parts of your body, and cause more tumors, more cancer, which is why if you have cancer in one spot, it may spread. And that is due to what we were talking about right here with this metastasis, this ability to move around. Angiogenesis means that they can form new blood vessels. So these cells, these tumors, can actually bring themselves nutrients and oxygen. That's a problem. That's how they grow. And again, nothing is going to come in and say, we're destroying you. There is no regulation and no apoptosis. The origin of cancer. Normal growth and tissue maintenance depends on a balance between the signals that promote and inhibit cell division. There are two types of genes that may be mutated when the balance is upset, and this could cause cancer. Oncogenes, or proto-oncogenes, code for proteins which promote the cell cycle in various ways. If one of these is mutated, it may become an oncogene. There are also tumor suppressor genes that code for proteins which inhibit the cell cycle and promote this apoptosis. If a tumor suppressor gene becomes inactive, it may promote cancer development. Both proto-oncogenes and tumor suppressor genes are normally regulated in coordination with the organism's growth plan. But obviously, if one or both of these is not being regulated, we can see cancer occur. if we see a tumor suppressor gene become inactive, that's a problem because these directly inhibit the cell cycle and prevent the cells from dividing uncontrollably. So we want that. Some tumor suppressors promote apoptosis. But if you have a mutation in a tumor suppressor and it causes the cell cycle, it can cause the cell cycle to accelerate. If your body isn't suppressing things properly, there's going to be an acceleration. Examples are the RB and the P53 gene, which both code for proteins with the same names. And I'm sure you've probably heard of the P53 genes somewhere. That's usually the one that's referred to when we talk about cancer, because if there's a problem with that gene, we usually see some big problems. So that's what the last line there says. Half of all human cancers involve an abnormal or deleted P53 gene, because then the regulation that we need is not taking place. Proto-oncogenes are normal genes which are part of the stimulatory pathway, and they promote progression through that cell cycle. They include receptors and signaling molecules. But if we see a mutation in them, they can become oncogenes. And these can specify an abnormal protein product or produce abnormally high levels of a normal product. We then see uncontrolled cell division resulting, and this can lead to tumors. And there are about 100 oncogenes which can lead to tumors. An example is the BRCA1, and mutations with this one is what we see causing breast cancer and ovarian cancer. Other causes of cancer. Telomeres. Chromosomes normally have special material at each end that are called telomeres. These get shorter with each cell division. When they are very short, the cell will no longer divide. Telomerase is an enzyme that maintains the length of these telomeres. But you could have a mutation in the telomerase gene. And this would cause the telomeres to continue to lengthen, and this could allow cancer to continually divide. So we just talked about eukaryotic cell division. We need to also mention prokaryotic cell division, because the prokaryotes, the bacteria and the archaea, do something a little bit different. The prokaryotic chromosome is a ring of DNA and just has a few associated proteins. It is folded up in an area called the nucleoid. So remember, the prokaryotic cell is set up differently than the eukaryotic cell. It doesn't have what we call that true nucleus. Therefore, it undergoes cell division a bit differently than the eukaryotic cell. There is something called binary fission, and this literally means the splitting in two. So two replicate chromosomes are distributed to two daughter cells, and this produces two daughter cells that are identical to the original cell, and this is called asexual reproduction. And there is a different time frame for each species. So E. coli, which is an intestinal microbe, can do this in about 20 minutes. It can reproduce every 20 minutes. Pretty quick. So when we compare the prokaryotics to the eukaryotics and talk about cell division, here are some main points. Binary fission and mitosis both ensure that the daughter cell is genetically identical to the parent. So they're both doing the same thing, but they are doing it differently. Prokaryotes, protists, which are algae or protozoans, and some of the fungi, which are yeasts, are single-celled. So cell division of a single-celled organism produces two new individuals. In multicellular fungi, plants, and animals, cell division is important for growth, renewal, and repair. During binary fission, the chromosome duplicates and each daughter receives one copy as the parental cell elongates and the new cell wall and membrane form between the daughter cells. And there is no spindle involved in binary fission. It's a different process. And that brings us to the end of this lecture on the cell cycle. I know it was a lot of information. Please review it and let me know if you have any questions. Have a fantastic day.